Jeotgalibaca is a genus of bacteria from the family of Carnobacteriaceae. Jeotgalibaca, a gram-positive bacterium, is found in traditional Korean salted and fermented food. It is made by adding 20 to 30% salt to various types of seafood. Sujo is a type of Jeotgalibaca, the name deriving from the Korean words seu, shrimp, and Jeotgalibaca. Among other seafoods, salted shrimp 25% with siwa. Tare and other ingredients are used for its preparation and subsequent fermentation. Salt-resistant aerobic and anaerobic bacteria exist in most Jeotgals, and recent studies have reported the isolation of many novel species and genera from this food. This also shows how it follows all members of its family of Jeotgals in salt resistance. 7 p less than 0.001 were found in our study. Aldosterone 18-glucuronide, an important metabolite of aldosterone, performs many important functions such as cell signaling, fuel and energy storage, and membrane integrity stability. Jeotgalibaca is used in panchin shrimp paste and in other pastes that are fermented from grasshopper subshrimps. Grasshopper subshrimps are found in the water that borders seawater and freshwater. The whole bodies are transparent, and the longest length are only 0.008 to 0.01 meters. Due to the tiny sizes of grasshopper subshrimp, shrimp pastes that are made from them tend to have lower fat and cellulose, aryl content, and higher astaxanthin and calcium levels than that made from other shrimps. Panchin is located in the southwestern part of Liaoning Province, the center of the Liaohe River Delta in China. There are 6 major genera of bacteria: Jeotgalibaca, Jeotgalicoccus, Lysinobacillus, Sporosarcina, Staphylococcus, and Psychrobacter that were found to be positively correlated with biogenic amine (SBA) production level, suggesting that these bacteria might have a strong tendency to produce BA. In recent years, there are some studies related to the impact of microbial diversity on BA formation in fermented food, such as fish sauce, sausages, and soy sauce. Cell morphology and features. This bacterium is heavily used in fermentation of some Korean foods, therefore is able to withstand environments of extremely high salinity. Cell morphology, as described from a primary source, found that Jeotgalibaca is gram-positive and consisted of non-motile, non-spore-forming coccus-shaped cells which are grouped into tetrads, sarcina, and conglomerates, phylogeny and genome evolution. The genomics and phylogeny of the Jeotgalibaca. We were able to find the components of the DNA composition of the bacteria and see how it compares to the other related phylogeny. Levels of DNA sequence similarity of the GRO-EL DNA contig, 465 BP, of strain EX07T were 81.3 to 82.8% when compared with the type strains of all species of the genus Trichococcus and less than 76% with the type strains of species of the genera Bavariacoccus and Granulicatella. Figure 1 shows the phylogenetic relationship between strain EX07T and its closest relatives based on GRO-EL sequences. The novel isolate formed an independent branch supported by a 100% bootstrap value and confirmed the moderate relationship with all members of the genus Trichococcus. The combined results of the 16S rRNA gene and GRO-EL sequence analyses support the classification of strain EX07T within a new genus. Metabolic activity Jeotgalibaca was tested under various environmental conditions, and it grows well on TSA plates. No growth was obtained on Na, PDA, and R2A agar. It is an aerobic microbe that gets carbon from sugars and organic acids, such as D-gluconic acid, and has chymotrypsin activity. The related species, Coriobateriaceae, phylum Actinobacteria, possess very similar metabolic processes to Jeotgalibaca. They have been reported to be involved in the metabolism of bile salts and steroids, as well as the activation of dietary polyphenols in the human gut. In particular, strong positive correlations between Coriobateriaceae and the steroid aldosterone 18-glucuronide, R = 0.